Welcome. My name is Madison Quintana, and I will be presenting my project on the distribution of hypocretin orexin and neuronal nitric oxide synthase in the hypothalamus and zona incerta of the adult male rat brain. This project was made possible with the funding of the Prell Scholarship, the University of Texas at El Paso, and with the amazing guidance of the Khan Lab. The hypothalamus is a brain region responsible for sleep-wake cycles, feeding behavior, and arousal. Although the cellular structures of this area have been explored, we lack a foundational understanding of the intricate chemical systems that compose this area. Chemoarchitectural research has been limited to a one-specimen representation. To ameliorate this, project aims to represent the projections of two neurochemical systems through 4 and 5 rats respectively on Swanson's Standardized Rat Brain Atlas, BrainMaps 4.0. The first neurochemical system to be explored is hypocretin 1 orexin A. This neuropeptide transmitter plays a role in feeding, wakefulness, and arousal. The second neurochemical system is neuronal nitric oxide synthase. This intracellular enzyme produces gaseous nitric oxide, which contributes to feeding and stress responses. Two methods were used to stain these sections, immunohistochemistry and a Nissl-based thionine stain. Sections were stained with the primary and secondary antibody for peroxidase immunohistochemistry to visualize immunoreactive HO and nNOS, while an adjacent series was stained with Nissl staining techniques to visualize the cytoarchitecture of these sections. These tissues were mounted, photographed, and then parcellated and mapped using Adobe Illustrator. Parcellated Nissl sections were then matched to their level according to BrainMaps 4.0 and to their adjacent immunohistochemistry stained section. This allows us to visualize immunoreactive cell bodies and axons onto the Standardized Rat Brain Atlas. As the neurochemicals were mapped across several brains, the data was averaged onto single-level high-density concentration isopleth maps, essentially a heat map for these chemical systems, and as well as visualized through the percentage of area that either HO or nNOS cell bodies and fibers occupied at each level. We can then visualize these densities in several fashions, such as the flat map of the neurite concentration of HO within the hypothalamic area as seen in Figure 3. Collectively, these high spatial resolution representations are novel for the scientific community. We are able to see where these essential cell populations are concentrated in the forebrain. These novel maps lay a foundation for future research by allowing scientists to target these populations for future experiments. I appreciate you joining this presentation and look forward to discussing any questions you may have. Thank you.